Operant conditioning, also called instrumental conditioning, is how we learn the outcome of behaviors or the consequences of our actions. This lecture is based primarily on material from Chapter 5 of our textbook, Learning and Memory from Brain to Behavior. We'll turn now to talking about the behavioral processes that are actually involved in operant, or as it's called, instrumental conditioning. And it involves a sequence of three things: a stimulus that discriminates, that tells the context in which the behavior is going to occur, the behavioral response itself, and the outcome. And there are a number of different ways in which behaviors can be modified through operant conditioning. Positive reinforcement is if something leads to a good outcome, you do it more. Okay, and that's in the previous example. Whenever someone did a good outcome, at least as Sheldon viewed it, then Sheldon would give them chocolate and they would do it more often, such as speaking in a lower register. So another example would be if you study, you get a good grade, you'll study more because a good grade is a good outcome. Positive punishment is if something leads to a bad outcome, you do it less. So that if you get punished by something, so if you arrive late, we take points off, you're punished, and therefore you'll arrive late less often. This is similar to the squirting of water in the face that we saw in the last scene. This was designed to make the person who was squirted do whatever they did just before that a little less often. Now here's a term which is frequently misused, misunderstood, and in fact Sheldon, in the last example, used this wrong. Negative reinforcement, in psychological terminology, is something which, if it ends or avoids a bad outcome, you do it more. Okay? Now Sheldon misunderstood this or misused it to be synonymous with punishment, but it's not. Negative reinforcement is a situation such as if you have a headache, you take an aspirin, a bad outcome, the headache ends, and therefore you take aspirin for pain more often. You're reinforcing the behavior of taking aspirin by removing a headache. Negative punishment is an omission. It ends or avoids a good outcome. You'll do it less often. So if you are in a game, you're enjoying the game, that's the good outcome. If you curse at the ref while you're playing at the game, he will eject you. This way, something good being in the game has been ended, and it followed your cursing at a ref, so you're going to curse at the ref less often. So really, this terminology is confusing. This is a graph that explains that if reinforcement is whenever a response is increasing, you're reinforcing that response. Punishment is whenever a response is decreasing. The positive and negative terms are very confusing. They refer to positive if you're adding it, negative if you're removing it, and it's just something that has to be learned correctly. Positive is added. It doesn't mean good. Negative is subtracted. It doesn't mean bad. If I had my way, I would change all the terminology, and we would call it instead additive reinforcement and additive punishment, subtractive reinforcement and subtractive punishment, so as not to be confused with the terms positive and negative. Here's another perspective on understanding this.
Many people confuse the terms negative reinforcement and punishment, thinking they mean the same thing, but they don't. Remember, reinforcers in general strengthen voluntary responses, and punishers weaken voluntary responses. So, growing up while I was in school, my mom could have punished me whenever I received bad grades in order to weaken whatever behavior was causing me to get bad grades. Or she could have reinforced my good behavior with something desirable whenever I did study or get good grades. Now, there are two types of each approach, positive and negative. Positive reinforcement is a process that strengthens the likelihood of a particular response by adding a stimulus after the behavior is performed. Negative reinforcement also strengthens the likelihood of a particular response, but by removing an undesirable consequence. So my mom could have told me that if I got good grades during the school year, then I wouldn't have to get a summer job, and that would be my reinforcer. Note that in both cases, the response of getting good grades is strengthened. We can punish a response by also adding or removing a stimulus. Like I could punish the bad behavior of a child by spanking its backside, or I could punish you after you speed by taking away your driver's license. Both of these things would act to suppress certain bad behaviors. So punishment is either adding or removing something that would stop a response from happening again. And reinforcement is adding or removing something that would encourage the response to happen.
Let's talk a bit about the stimulus and what that role that plays. It's a discriminative stimulus or a context. It tells you whether or not these contingencies are operating, namely that if you do a certain behavioral response, a particular outcome will occur. But these contingencies can change. For example, if I cry, mom picks me up, as seen here, I being a baby. But what if mom only does that when she's sober? If I cry and she's drunk, she does nothing. So in this case, whether mom is sober or drunk is a discriminative stimulus, which tells us which contingencies are in effect, whether or not crying will or will not lead to me being picked up. Operant conditioning is thus a three-part association. There's the context or the discriminative stimulus, S, the behavioral response, and the outcome. The discriminative stimuli are crucial in this type of learning, ensuring that we select behaviors that are appropriate to the situation. For example, you would only put money into a soda pop machine that's working. If it's not working, it's broken. If it's not plugged in, you wouldn't put the money in. Rats can be trained to press the lever for food, will learn the sound of the equipment being turned on, and will only press the lever when it's actually functioning. To see if you've understood the principles of S, R, and O, here are a couple of examples where I'd like you to identify for yourself, test your understanding for this SRO. So back to the idea of a baby's perspective. Here's a cartoon, High and Lois. First panel, the baby says, "This is great." We pan backwards and we see the baby's in the middle of the parents' bed between them, saying, "I'll have to wake up crying in the middle of the night more often." So from the baby's perspective, we're talking about conditioning, operant conditioning of the baby. What's the stimulus, the response, and the outcome? The answer is the stimulus, or the context, is the baby is lonely in its crib. It learns that if it responds to this loneliness, it gives a response of crying, then the outcome is it gets brought into bed and gets sleeping with its parents. It's no longer lonely. So we can take this same cartoon, the same situation, and look at it from the parent's perspective. So now how are the parents being conditioned through operant conditioning? What's the S, the R, and the O? Here, the stimulus that they hear is they hear the baby crying. Their response is they bring the baby to bed. The outcome is the crying stops. So an aversive stimulus, namely the hearing the crying, is reduced by bringing the baby to bed. Here's a not-so-typical rat experiment. The rat is on the phone saying, "Oh, not bad. The light comes on. I press the bar. They write me a check. How about you?" So in this cartoon, what's the stimulus, the response, and the outcome? The stimulus is the light. The response is the press, like in the rat experiments we were seeing before. But here the outcome, rather than being a sugar pellet, is that the rat is paid by a check. Our understanding of these types of situations, the operant conditioning or instrumental conditioning, owes a lot to Edward Thorndike, who developed what he called the law of effect, and his research was based on studying cats' trial and error learning to escape a puzzle box. We can take a visit to his home laboratory in the following video.
But how is a new skill learned? That was a question which began to fascinate Thorndike. To answer it, he built some ingenious puzzle boxes from which cats could only escape by operating latches.
And in you go.
The cat appears to be very clever in engineering its escape, solving the problem with a deftly placed paw and a push of its nose. But Thorndike didn't believe that an animal, even a clever cat, understands the consequences of its behavior. When he placed a cat in the puzzle box for the first time, Thorndike was unable to see any evidence of flashes of insight. The successful actions appeared first by chance. He proved that the apparent cleverness arose by trial and error, and used graphs to measure the rate of learning. A well-practiced cat quickly recalls the actions that help it escape to its reward of food. If an action brings a reward, Thorndike believed that that action becomes stamped into the mind. In his thesis, he explained further his ideas about learning, that behavior changes because of its consequences. He called this his law of effect, which explained how even wild creatures develop new habits.
Review what we saw here. There's the cat puzzle box and the learning curve showing the time to escape. Initially, lots of behaviors are tried out. It's essentially exploring the environment. The animal tracks the outcomes of behaviors. In context S, namely this box, a particular response produces the outcome O, which is getting out of the box. This knowledge guides future behavior, which is what Thorndike called the law of effect, namely behaviors with positive outcomes and good outcomes increase, behaviors with bad outcomes decrease. Puzzle box had some methodological problems. You'd have to repeat trials over and over, resetting the animal and the device, and it's hard to compare across animals, which is why most conditioning research these days or since then used a different approach developed by B.F. Skinner.
Can pigeons read? This one gives every indication, because he's been taught to distinguish between two words and to behave appropriately. He's learned his different response to each sign by being rewarded with food. So the bird isn't acting independently. Its behavior is shaped by controlling its environment. The first task was to isolate an individual piece of behavior and see how that could be changed. Skinner did this by keeping individual pigeons at about 3/4 of their normal weight, so that the birds were always hungry, and food could be used as an automatic reward. The pigeon was studied in a uniform box, one it quickly grew used to. One piece of behavior, pecking a coloured disc, was measured on a graph. The pigeon learned that pecking the disc produced a reward. Then the behavior of pecking could be studied in relation to how often that reward was offered, or in Skinner's terms, what was the schedule of reinforcement.
The main thing is what we call schedules of reinforcement. Reinforcement is what the layman calls reward, and you can schedule it so that a reward occurs every now and then when a pigeon does something. We usually use a response with a pigeon pecking a little disc, a little spot in the wall, and you can reinforce with food, but you don't reinforce every time. Perhaps every 10th time, or perhaps only once every minute or something like that. There are a very large number of schedules, and they have their special effects. And there is a good example of how you can move from the pigeon to the human case, because one of the schedules which is very effective with rats or pigeons is what we call the variable ratio schedule, and that is at the heart of all gambling devices and has the same effect. The pigeon can become a pathological gambler just as a person can. Now, the fact that we found that out with pigeons and could prove it by removing and changing the schedule makes it easy for us to interpret the case with the human subject. We don't say that the human subject gambles to punish himself, as the Freudians might say, or gambles because he feels excited when he does so. Nothing of the sort. People gamble because of the schedule of the reinforcement that follows, and this is true of all gambling systems. They all have variable ratios built into them. So what we've learned from the pigeon, it made it possible to interpret this vast field very effectively.
Where does that leave free will? Because we all think we have a choice whether to do things or not to do things.
Yes. Well, you see, it leaves it in the position of a fiction. We have assumed somehow or other that these internal states, feelings, and so on, have initiated something. They have started something. They have created. We have done something in a voluntary way. We have willed to act. If you now look at the actual history, we find that there are external reasons why this has happened. In other words, by discovering the causes of behavior, we can dispose of the imagined internal cause. We dispose of free will as an American divine of the 18th century, Jonathan Edwards, did. He said, we believe in free will because we know about our behavior, but not about its causes. And of course, it's the object of a science of behavior to discover causes. And once you have found those causes, there is less you need to attribute to an internal act or will, and eventually, I think you need to attribute nothing to it.
Skinner box that you saw in the last video. There's a box, there's an animal, a rat, it has some food, there's a lever it can press, and a reservoir. The outcomes are food delivery shown here, or a shock that can come through the wires. The behavior is the rate of lever pressing, and the context is a light that signals the box is on and that the contingencies are relevant. Note the animal is free in the chamber. There's no experiment or intervention. This is why it's often called free operant paradigm. And many contingencies can be introduced. Just to review the sort of micro world, the positive reinforcement is you get food. The negative reinforcement is it ends shock. So this it's increasing behavior by removing something that's aversive. Punishment is you get shocked, therefore you will press the lever. Negative punishment, if you press the lever, the food stops, so you stop pressing the lever. So let's examine learning in more detail in a positive reinforcement paradigm where the rat is pressing it and getting food, and therefore the more it presses, the more food it gets. So the animal is really operating on the world here. It's important to compare this type of learning to the classical conditioning we talked about in a previous lecture. So let me compare them point by point. Animal operates on the environment in the operant conditioning, and in the classical conditioning, the environment operates on the animal. The animal is learning about the environment, but it's not affecting the environment. The stimulus evokes a response to produce an outcome, SRO, pathway in operant conditioning. In classical conditioning, the stimulus evokes a response. It's just an SR pathway. An animal connects the context, behavior, and outcome in the three-way link. In classical conditioning, it's a two-way link. The animal learns that the CS predicts a US. So to summarize the behavioral processes, animals adjust their behaviors by tracking the consequence of their behaviors in a stimulus-response-outcome arc. The consequence determines future behavior. Positive reinforcement produces a pleasant outcome. You increase the behavior. Negative reinforcement removes a noxious outcome. It increases the behavior. Punishment produces a noxious outcome and decreases the behavior. Omission or negative reinforcement removes a pleasant outcome and therefore decreases. So the response learned is an overall goal or intention. It's not just a simple rote behavior. However, the rote behavior actually improves with practice. Animals are sensitive to the context in which consequences occur. A discriminative stimulus or context tells when a response-outcome relationship exists. So, for example, on the potty, peeing is rewarded. If a baby is not on the potty or a toddler is not on the potty, then peeing is not rewarded. What are the brain substrates of these behaviors? Well, there are two areas that are particularly important here. The dorsal striatum, which plays a role in linking the stimulus with the response, the SR pathway, and the orbitofrontal cortex, which plays a role in linking responses with outcomes, the RO arc. The dorsal striatum can be shown here in this graph, and it's connected by dopamine pathways below, the substantia nigra, which sends dopamine to the dorsal striatum, and also the ventral tegmental area, which sends dopamine to the frontal cortex and other areas of the brain. The basal ganglia are a set of subcortical structures that link sensory and motor cortices. Within the basal ganglia, the dorsal striatum comprises the caudate nucleus and the putamen, and as I noted, it seems to play an important role in SR learning. Let's take a closer look at the ventral tegmental area, which is going to be critical for our discussions moving forward.
The ventral tegmental area, or VTA, is found in the midbrain, situated next to the substantia nigra. Although the VTA contains several types of neurons, it is primarily characterized by its large population of dopamine neurons. It is one of the two major dopaminergic areas in the brain, the other being the substantia nigra. VTA dopamine neurons travel from the VTA to other areas of the brain in several major pathways. Two of the most prominent pathways are the mesocortical and mesolimbic pathways. The mesocortical pathway projects from the VTA to widespread areas of the cerebral cortex, including the prefrontal, orbitofrontal, and cingulate cortices, as well as sensory and motor cortices. The mesocortical projections are very diverse, and the pathway is involved in a wide range of functions, including motivation, emotion, and executive functions. The mesolimbic pathway projects from the VTA to several limbic structures. The largest projection of this pathway is to the nucleus accumbens, but other projections stretch to areas like the amygdala as well. The mesolimbic pathway also has diverse functions, but it is most frequently associated with the reward system, as dopamine signaling along the mesolimbic pathway is considered to be important to the processing of rewarding experiences. When someone uses an addictive drug, for example, dopamine neurons in the VTA are activated. These neurons project to the nucleus accumbens via the mesolimbic pathway, and their activation causes dopamine levels in the nucleus accumbens to rise. The effects of these increased dopamine levels are still not fully understood, but they may be involved with encoding memories about rewarding experiences and attributing importance to environmental stimuli that are associated with the reward. Called the basal forebrain, which is located near the front and bottom of the brain. The nucleus accumbens is the major component of the ventral striatum and is situated between the caudate and the putamen. The nucleus accumbens is typically divided into two anatomical components, an outer shell and a central core. There are thought to be functional differences between these two regions, where the shell is more associated with the limbic system and the core is more strongly connected to the motor system. It should be noted, however, that while this distinction between shell and core is clearly seen in rodents, it is less evident in humans. Although the nucleus accumbens is best known as part of the reward system, its functions are much more complex than simple reward processing and are still not fully understood. The nucleus earned its reputation as a key part of the reward system in a large part due to its connections with the ventral tegmental area, or VTA. Dopamine neurons project from the VTA to the nucleus accumbens as part of the mesolimbic dopamine pathway, and this pathway is activated in association with rewards. However, the exact role of the nucleus accumbens in processing rewards is not completely clear. It is thought that the nucleus accumbens likely plays a role in learning about rewards and the stimuli that are associated with them. It also seems to be important to stimulating the pursuit of rewards and the selection of actions that are most likely to result in the attainment of a reward, along with the suppression of actions that are less likely to be useful. The nucleus accumbens also appears to be important in processing aversive experiences, however, and in learning to move away from aversive stimuli. Thus, the nucleus accumbens appears to be involved in responses to all motivationally relevant stimuli, whether rewarding or aversive.
Back to the dorsal striatum and its role in stimulus response learning. In rats, lesions to the dorsal striatum produce problems with operant conditioning. The simple response-outcome relationships are learned correctly, press the lever, get the food, but no learning takes place with the discriminative stimuli, such that a rat is unable to learn that if the light is on, then and only then press food. This shows a specific role of the dorsal striatum in linking discriminative stimuli to behavioral contingencies. This figure shows that instrumental learning may involve the interaction of several neural systems. The visual cortex brings in the sight of a lever, sight of other stimuli. The motor system is where the response is activated. This leads to a consequence of food, which affects the taste system. It affects the hunger system, which then turns to the reinforcement system, which either increases or decreases one's potential for responding. One way of studying these reinforcement circuits is through electrical brain stimulation. One of the pleasure centers of the brain is the ventral tegmental area in the brainstem. As we saw before, it's the center for dopamine neuromodulation. VTA stimulation is by itself a powerful reinforcement. Brain stimulation may directly activate the brain's reinforcement stimulus, as shown in this diagram below, eliminating the need for natural reinforcers. What happens if you put an electrode into somebody's pleasure centers, into the VTA or perhaps the nucleus accumbens? We'll see in the next slide some historic video from some of the earliest experiments that were done that looked at electrical stimulation of the brain.
You almost look like you're smiling a bit. Are you?
Oh, no, I might.
Heath implanted electrodes in the brains of chronically depressed patients, hoping to stimulate pleasurable sensations.
I guess I've cracked up all the way. I don't know.
I mean, that's the reason you're smiling.
Probably.
Well, you look like you're really... What are you laughing about?
I don't know.
Huh?
What are you doing? Are you doing something to me?
Not surprisingly, the experiments were controversial, but the electrodes did have an effect, especially near an area of the brain called the nucleus accumbens. Heath even let his patients control their own stimulation.
I find this button the best.
That's number 2 button.
Most pleasurable.
Mm-hmm. How do you feel?
I made a small joke yesterday. I don't know if I should repeat it or not.
I'd like to hear it.
Do you think it's worth repeating? What about the very deep electrode all the way down?
You're trying to tell me how it excites you, is that it?
Oh, I think it's somewhat of a sexy button.
Yeah. Is that why you said it went all the way down?
Perhaps we get pleasure from drugs because they stimulate the same brain areas as sex and food. Working in Montreal in the 1950s, James Olds devised a crucial test. He worked out a new way to, in effect, ask animals how they felt.
This is a device which trains the animal to turn on the stimulus for himself. Here you notice the animal wandering toward the pedal. When he first touches it, he gets no stimulation. It's not connected yet. Then, one time he touches the pedal, it does turn on the stimulation. You can see the lights go on.
Now, the interesting thing about the old stimulation, the rewarding stimulation, was the strength of it. It was somewhat different than rewarding stimulation for food and water in that there was no satiation. Animals would continue for hours and hours and didn't seem to satiate. They didn't feel full. They would just keep doing it for long, long periods of time. They would do it to exhaustion.
Now, I will electrify the grid. The animal must cross an area that gives a very painful shock to the feet in order to get to the pedal and stimulate the brain.
And a rat wanting brain stimulation would brave a shock stronger than even a starving rat would brave to get food.
This shows a rat is willing to pay a very high price in order to get to the pedal.
Human addicts will pay just as high a price for their rewards.
The fascinating thing was that we found in our laboratory that every single drug that increased sensitivity of the animal to brain stimulation was either an abuse substance or a substance that has potential for abuse.
So James Olds' technique of the 50s gave scientists a way of testing for the addictive potential of drugs. And a way to find out where in the brain they acted.
Now, I think there's general agreement that this is the area where the action is in terms of the rewarding effects of drugs. Whether they be heroin, whether they be amphetamine, whether they be cocaine or angel dust or whatever, any drug that produces rewarding effects, it's believed this is where the action is.
Using similar methods, Chris Vivitrol and his colleagues in Vancouver wanted to know which pleasure areas are active when we crave both drugs and food. They found that the area Heath stimulated in his patients, the nucleus accumbens, is the center of a key network, the dopamine circuit. Heroin seems to act in one way. Cocaine in another. And both hit the nucleus accumbens, which the Canadian scientists recently proved is also active when we crave food.
If we selectively take these dopamine neurons out of that part of the brain, the animal loses interest in self-administering cocaine, it seems. Apparently, the drug is no longer reinforcing. The same story seems to apply for amphetamine. If you destroy these dopamine-containing neurons in the nucleus accumbens, amphetamine self-administration disappears as well.
A heroin addict has volunteered to be given a large dose of morphine, chemically similar to heroin, by scientists at the National Institute for Drug Abuse in Baltimore.
Relax.
Today, using new methods, researchers can at last start to look at where drugs act in the human brain. The new machines reveal why different drugs make us act like primitive animals. Morphine shuts down the higher cortex and leaves the older emotional brain in charge. Cocaine gives a boost to the whole brain, but especially stimulates the primitive centers of emotion. So from those rough and ready experiments in the 1950s, scientists did learn where in the brain many addictive drugs act.
The role of dopamine and reinforcement. Some VTA axons extend to the nucleus accumbens in the basal ganglia. The nucleus accumbens sends dopamine then to motor areas in the striatum. Dopamine in this way may be the physiological basis for the wanting aspect of reinforcement, that is, the motivation or wanting in chemical form, and this is what contributes to addictive behavior. One way to see this is to look at what's called the incentive salience hypothesis, the idea that dopamine motivates learners to work for reinforcement. In the experiment that took place, that I'll show you the data in a second, both control rats and rats that are given a dopamine agonist prefer sugar pellets over rat chow. Generally, all rats prefer sugar pellets over rat chow, and if both are freely available, whether they have a dopamine agonist or not, they'll both choose the sugar pellets. But if you introduce a small work requirement for the preferred food, this changes. Given a choice between lever pressing for sugar pellets or eating freely available but less appealing rat chow, regular control rats will press the lever for the sugar pellets. They'll execute a high number of lever presses to obtain the pellets, and they'll largely ignore the chow. Rats given the dopamine antagonist will press the lever much less frequently and eat much more free chow, even if they like it less than the sugar pellets. So we see here to work by lever pressing for sugar preferred food or sugar pellets is very high in the control rats, but those who have dopamine disrupted are not. In contrast, those where there's the free consumption of less preferred chow food, the chow, when sugar is available via lever pressing, the control rats will show very little eating of the free chow, but those with the dopamine disrupted will prefer the free chow. They won't bother to work for it. So it suggests that disrupting the dopamine pathways prevents animals from being able to make a behavior that leads to a future consequence, a future good consequence, even if they were still interested, even if their preferences, the degree they like the sugar pellet or the other pellet, has not changed. So this brings up an important distinction between wanting and liking in the brain. Many researchers believe that we have separate brain systems for signaling hedonic value, meaning the subjective goodness of a reinforcer or how much we like it, and these are distinct from those signaling a motivational value, meaning how much we want a reinforcer and how hard we are willing to work to obtain it. These endogenous opiates are naturally occurring neurotransmitter-like substances that have many of the same effects as opiate drugs. Endogenous opiates are released in response to primary reinforcers, such as food, water, sex, and they may be released in response to secondary reinforcers and pleasurable behaviors too. So in this way, the endogenous opioids, which are what are activated by opiate-like drugs like heroin, are directly approaching the liking aspect of the brain, in contrast to the dopamine drugs, which are affecting the wanting. Another perspective on understanding the role of dopamine in reward is work done by Wolfram Schultz, who trained monkeys to press a lever for food. In his electrophysiological recordings, indicated that these dopamine neurons in a monkey's midbrain signal reward, expectation of reward, or omission of reward. And we'll turn now to let Wolfram Schultz describe these classic experiments himself.
Give the animal a drop of liquid at the mouth repeatedly, like 20 times. Repeatedly, we see that every time after that reward, drop of liquid, grape juice or something, we get a few more extra action potential. Not a whole lot, but there is about 100,000 of these neurons on each side in the brain in a monkey, and about 80% of them show that response. So it's quite a nice volume, big response that lasts maybe 200-300 milliseconds. If we give a stimulus that predicts that reward, then we don't get a response here anymore, and the response moves over to the stimulus, which is kind of funny. And if you're a little obsessed like us and like to play in the laboratory and got nothing better to do, then of course sometimes you won't give a reward here, and then you see that the activity of this neuron is depressed. Right? And the response still stays there because the stimulus predicts the reward, but you're withholding the reward by clamping the liquid line or something, or the animal making an error and not getting a reward. Right? So that is a bit suspicious, and then we kind of made a little calculation and realized that the response of these neurons are governed by the difference between the reward they're receiving and the reward that is predicted. So here nothing is predicted in time, nothing is predicted, and the reward comes out of nowhere, bam, suddenly a drop of liquid. Here, the reward is predicted by the stimulus 1.5 seconds before. The reward comes, everything is predicted, you get nothing. And here, the reward is predicted, right? You get a prediction back here, but you get no reward. So this equation then is negative, right? Rescorla-Wagner called that the associative strength, the difference in associative strength actually had a even more complicated word, which was totally hopeless to pass across an audience that wants to have a straight explanation of these very surprising data that nobody had seen before. We called it unpredictability in a paper, and then came, of course, the idea with the analogy with the reinforcement models, and that nailed the term of reward prediction error. Now, this is really terrible, because reward to prediction error means that you're making an error and you're getting a reward. That's what my colleagues tell me. It's bad. But the idea is that this reward prediction error, lambda minus v, is the crucial learning term in these asymptotic learning theories like Rescorla-Wagner or temporal difference learning, and this is exactly what these neurons are coding. So the idea of prediction error comes from this general circuit diagram of error-driven learning that you may have seen when you looked at control theory at undergraduates, right? You start out with a prediction, you get some outcome, you compare the two, maybe they're different, then you say, well, that occurs when you made an error, or it's actually the error is simply the discrepancy between these two terms, and then you update the prediction with this error. Next time, hopefully, your prediction is a little bit better, and then the prediction error gradually becomes smaller and smaller. And as you change your prediction, of course, you change your behavior, right? That's why it is called learning. And then after a while, if the prediction matches the outcome, note that the outcome doesn't change, the prediction gets adapted, right? Then you don't change the prediction any longer, because if you're right on target, you get your reward. You won't change anything in your behavior. You just keep pressing the same button to get the same can of Coke, okay?
This is a study of reward prediction by dopamine neurons. In the study, dopamine neurons in a monkey's midbrain respond strongly after unexpected rewards. If a light occurs before a food, dopamine neurons increase activation after the light if the light has been repeatedly paired with the food, but not after the food when it's expected. And the dopamine neurons decrease activity after an expected reward does not occur, namely emission. This illustrates what Schultz referred to as the reward prediction hypothesis. It comes out of the Rescorla-Wagner model, which you saw in the previous lecture on classical conditioning, and it suggests that dopamine is involved in predicting future reward using the same kind of error-correcting processes described by Rescorla-Wagner. Here again is the figure that Wolfram Schultz showed. There's in an unexpected juice award in a naive animal, there's dopamine firing after the juice. When the reward is predicted by a light stimulus, so there have been many light-juice pairings, the dopamines fire after the light, but not after the juice. When you have in a trained animal, when the light is presented and there's no juice, then you see a decrease in firing, suggesting the expectation has been failed to appropriately predict what it was expecting. So we've seen here several theories about the interaction of dopamine and reinforcement. The incentive salience hypothesis suggests that dopamine modulates wanting rather than liking, namely how hard an organism is willing to work for reinforcement. The reward prediction hypothesis argues from Schultz's work that dopamine signals whether reinforcement is expected. These are not necessarily alternative hypotheses. They're different ways at looking at different bodies of data and suggest some of the many ways in which dopamine is involved in reinforcement. This leads us next to talk about the clinical applications of operant conditioning, of the neural substrates of operant conditioning, what happens when they go out of whack. And as suggested by some of the earlier discussion, the most common way in which the dopamine system is disrupted is through drug addiction. Pathological addiction is a strong habit maintained despite harmful consequences. This involves craving a high euphoria and avoiding withdrawal. Seeking pleasure involves positive reinforcement. Avoiding pain involves negative reinforcement. And although liking a drug may help initiate addiction, the incentive salience hypothesis suggests that addiction is maintained by wanting a drug, so that across the time course of addiction, initially a drug may lead one to get high, and therefore there's a hedonic value, but ultimately what keeps an addict coming back to the drug isn't the fact that it makes them feel good, it's that they have a compulsive wanting for the drug, and the absence of the drug produces a desperate craving or wanting for it. All addictive drugs cause the release of dopamine from the ventral tegmental area. We can understand thus the if all of them are increasing dopamine, different drugs affect this increase in dopamine in different ways. We see here the difference between amphetamine and cocaine. So on the left just shows a standard terminal. There's a presynaptic dopaminergic neuron. There are little vesicles in one which can pass across the synaptic cleft and connect to dopamine receptors at the postsynaptic terminal. Amphetamine increases the amount of dopamine that's released. That causes more dopamine, while cocaine interferes with dopamine reuptake, the recycling of dopamine, and therefore cocaine allows more of the dopamine that has been released to make it across to the postsynaptic. The net effect in both cases, whether it's amphetamine or cocaine, is more dopamine is hitting the postsynaptic neuron. What can we do about people who become addicted? Well, there are two main approaches: the cognitive and behavioral therapies involving varieties of extinction and distancing and reinforcement of alternate behaviors. Ironically, just as the dopamine system is the basis for instrumental operant conditioning, the methods for treating drug addiction also work by tapping into instrumental conditioning. But recent studies have shown that you can actually enhance the withdrawal from an addictive drug by using other dopamine drugs. Naltrexone is a drug which indirectly inhibits dopamine production, and it's been shown to help treat heroin addicts as well as compulsive gamblers, as we see in the next video.
The next medication is naltrexone. This is kind of confusing because it comes in different versions, and you also have naloxone, which is Narcan. But naltrexone is a drug that blocks your opiate receptor. What does that have to do with alcohol? Because when you drink and you get that high feeling, that is mediated by your opioid receptors in your brain. It's the same pathway as using opioids. So if you block your opioid receptors, you don't enjoy drinking. And it's been shown to work with pills. It only works in research studies because if you take the pill and you're just deciding, well, you know, this is Super Bowl weekend, I'm not going to not drink, so I'll just stop taking my pills. And people stop and they never start again, so the pills by themselves really don't work. What does work is the shot. Vivitrol is an injection of monthly naltrexone in oil, and it's been shown to decrease drinking, both in terms of percent abstinent and heavy drinking days. And if you look at the study on that arrow you see on the right, it shows that people who got the high dose shot at 6 months, about 30% of them were abstinent. And you may think that 30% is not a good number, but in medicine, anything over 15% is considered to be successful. And the placebo group who didn't get any naltrexone, at 6 months, about 10% of them were abstinent. So they went from 10% abstinent to 30% abstinent, and again, in medicine, that is considered to be a successful result.
Perspectives. Addictive drugs, heroin or caffeine, both legal and illegal, hijack the brain's reinforcement system, and treatment for people with addictions may include both medication as well as behavior therapies, which are based on principles from instrumental conditioning. This brings us to the end of the content of this lecture. What should you have learned from this? What are you responsible for in the exam? Well, from the behavioral processes, you should be able to explain how Thorndike discovered operant conditioning, identify the components of the learned association stimulus-response outcome, understand the similarities and important differences between classical and instrumental conditioning, and be able to identify whether classical or operant conditioning is occurring. Be able to distinguish between reinforcement and punishment, define and identify the role of discriminative stimuli in operant learning, define and differentiate the processes of positive reinforcement, positive punishment, negative punishment, also known as emission, and negative reinforcement. For brain substrates, you should be able to describe the role of the basal ganglia in operant learning, describe the neural circuits associated with reward in operant learning, describe how dopamine circuits in the brain are involved, explain how the ventral tegmental area is involved in reinforcement, and explain the role of opioids in reinforcement. From clinical perspectives, you should describe the role played by operant learning in drug addiction, describe the brain circuits affected by certain drug addictions, and describe the treatments used for drug addiction. So thank you. If you'd like some little bit more reading about applications of operant conditioning, here's some optional reading.